we're going to start the program. Only if you just take the charge. Yeah.
Of course, like Deshmukh sir would be the first person to allow us to move forward.
Yeah, sure.
Yes, you proceed. No problem.
Okay, so good evening, everybody. So we have gathered today online for this national seminar and students poster competition in commemoration of National Cancer Awareness Day 2024. The theme of our program today being Early Detection Saves Life. The event is being organized jointly by Department of Microbiology and IQAC of Raidiki College, Dhrubochand Haldar College, Michael Madhusudan Memorial College. All three colleges are from West Bengal in collaboration with Microbiology Society India. We are honored to have with us today our resource person, Dr. Mrinal Kanti Ghosh. Sir, welcome. Our chief patrons are Dr. A.M. Deshmukh Sir, All India President, Microbiology Society India. Dr. G.M. Helaluddin, Principal, Michael Madhusudan Memorial College. Dr. Shashobindu Jana, Principal, Raidiki College. Dr. Shottobroto Shahu, Principal, Dhrubochand Haldar College. We are extremely grateful to all of them. Joint conveners of the event are Dr. Kirat Kumar Ganguly, State President, MBSI, West Bengal Unit, IQAC Coordinator and Assistant Professor, and Head of the Department of Microbiology, Michael Madhusudan Memorial College. Dr. Malini Basu, MBSI State Coordinator, West Bengal Unit, Assistant Professor and Head, Department of Microbiology, Dhrubochand Haldar College, and myself, Dr. Urunima Bishwash, MBSI State Coordinator of West Bengal Unit, and IQAC Joint Coordinator and Assistant Professor, Department of Microbiology, Raidiki College. So, now I'll hand it over to our West Bengal Unit State President, Kirat Sir, to formally welcome everybody to today's event. Kirat Sir, please. Kirat, please unmute.
Yes, am I audible?
Yeah.
Yes, now you are. Yeah.
Yes.
Okay. It's my great pleasure to welcome you all in the national seminar and student poster competition in commemoration of National Cancer Awareness Day today, 7th November 2024. We are gathered here today not only to enhance our understanding in cancer and its treatment, but also to celebrate the innovative spirit of our students in the field of microbiology and its application in cell and cancer biology. All our activities for our students, our beloved students, and for other parts of the society. This seminar is organized jointly, as mentioned, jointly by the Department of Microbiology and IQAC of Raidiki College, Dhrubochand Haldar College, and Michael Madhusudan Memorial College in entire collaboration and support from Microbiology Society of India. In this connection, I would like to express my gratitude to Dr. A.M. Deshmukh, without whose enthusiasm and support, we couldn't be able to proceed a single bit in this type of programs. Dr. A.M. Deshmukh, President of Microbiology Society India, Dr. G.M. Helaluddin, Principal, Michael Madhusudan Memorial College, Durgapur, Dr. Shoshibindu Jana, Principal, Raidiki College, Dr. Shottobroto Shahu, Principal, Dhrubochand Haldar College. We are honored to have with us Dr. Mrinal Kanti Ghosh, renowned expert in cancer biology and inflammatory disorder and overall cell biology. The knowledge of Sir will be reflected in this platform today. It is a great pleasure for us to host a scientist of that stature in this platform today. He will share his invaluable insights with us today. As we delve into the theme of posters mainly, the microbes and microbial products in cancer treatment, recent insights, I encourage all the participants to engage actively by submitting poster, ask questions, and share your thoughts. This is a wonderful opportunity for us to learn from each other and to explore the critical role that microbiology plays in cancer research and treatment. So this idea is actually dissemination of knowledge from a great scientist and engaging the student for the application of microbiology in cancer biology. I'd like to extend a warm welcome to all the students participating in the poster competition, who will be participating in the poster competition, for which the guidelines will be shared in our Telegram group very shortly. Your hard work and creativity are commendable, and I took forward seeing innovative ideas from you on your posters. Let us make this seminar memorable and enriching experience for everyone involved. Thank you, and let's get started. It's over to Dr. Arun Vishwas.
Thank you, Kirat sir. And now it's time to welcome Dr. A.M. Deshmukh sir, All India President, Microbiology Society of India, without whose inspiration this entire society will probably not be possible. So Deshmukh sir will request you to say a few words for today's event. Sir?
Thank you. Thank you, madam. I'm here today to appreciate the efforts of Microbiologist Society Chapter, West Bengal. West Bengal is far ahead in microbiology, and the activities are conducted, organized by many hard worker, enthusiastic teachers. Means I can take the name right from Mukherjee ma'am, Swapna ma'am, Biprana sir, Kirat sir, Arunima ma'am is there, and many other teachers. So some teachers have organized regular uh, online lectures. So like that, many teachers are doing excellent job in microbiology. And today, the lecture on a very burning topic. But why I'm calling burning topic? Because the population of the world is 800 crore. And among them, about 8 million people are dying every year throughout the world because of the cancer. So it's a very burning topic. And Dr. Ghosh will focus the light on this topic. So I'm really, because and Kirat sir has told me, this lecture is being recorded. So very nice, we'll circulate it again. And many other students and teachers which are unable to attend today, they will watch this lecture. And our main goal, main motto is to percolate the knowledge, to popularize the knowledge, and to make the people aware regarding the cancer. So today I will appreciate Dr. Kirat sir and his team. They have done excellent job, and I expect such more many lectures in the future which will create awareness about the microbiology. As a president of Microbiology Society, it's my duty to wish you all the best and to welcome Honorable Dr. Ghosh as a speaker today. Thank you very much.
Thank you, Deshmukh sir. And now we'd like to invite Dr. Sanjeev Patankar sir, another stalwart and inspiring figure from MBSI, who was our former national coordinator. And currently he is the coordinator for courses in the Ayush Ministry on behalf of MBSI. Patankar sir, we invite you to say a few words, please.
Good evening to all of you, and I am very much thankful to organizers of this particular webinar and inviting me to participate in this. No doubt, even there won't be any invitation, I would have joined, because the topic is such that the it is the it is a very burning issue, and the person who is giving the lecture, he is a stalwart and he is the authority in a particular field. So uh, uh, at the outset, all the principals of the three colleges, the Michael Madhusudan College, Raidigi College, and the third one, that is the Drupchand Haldar College, they are so they are also taking very keen interest in this particular such type of the activities. They are supporting the Microbiology Society India for the different type of the activities as such. So because in past also I attended certain type of the programs for the Michael Madhusudan College. I have personally visited Drupchand Haldar College last year. So all these colleges, they are supporting the Microbiology Society India and for their work. So it gives a good type of the sign that the academic environment that exists in all these colleges and as such in the West Bengal. So again, I'm not taking much more time. The today's event, it is a very, very important, as Dr. Deshmukh sir said, and the theme of this particular type of the webinar, it is what the early diagnosis saves the life. So this is very, very important requirement in the cancer and the cancer biology. And then again, the topic of the today's lecture, it is the integrating biology, cancer in 21st century, integrating biology and emerging technologies, a holistic approach. So this is what it is needed in order to tackle the problem, because all of us know the cancer patients, they are increasing, and as of now it is nearly about every year new 41.1 lakh people, they are suffering from the cancer, particularly in case of the breast cancer and HPV. The increase in the patients, it is very, very high. In case of the ladies, they are suffering this particular type of the issues. HPV vaccination, we always promote, or we always awareness, give the awareness for the HPV vaccination also, which is available nowadays. One thing it is there, there are certain microbiological interventions, they can also be done. For example, the promotion of probiotics and other type of the thing, because they are also anti-cancerogenic, and they are having such type of the properties. Then regarding the nutrition and diet, everyday diet, because of the lifestyles, changed lifestyles, the particular type of the effect it is coming, environmental effects, that is the epigenetic effects, they are all contributing to this particular type of the phenomena. So this is a very, very important type of the topic, where the microbiology, biotechnology, all of them, they have to play a key role, a vital role in the diagnosis. Early diagnosis, it is the need, because if it is diagnosed early, the persons can be saved. Lot of research it is going on which Dr. Mrinal Kanti Ghosh, he will throw a light, and he will give the his expertise and his views on the particular area. So I once again congratulate you all for organizing this particular lecture, and always the Microbiology Society India promote such type of the activities, and always we see that where the microbiological interventions which will give the certain type of the relief and the positive effects on the society. So thank you for calling me and giving this chance. So I wish you all the best. Thank you once again.
Thank you, Patankar sir. I'd like to mention here that our principals from all the three colleges, although they couldn't be here, they have all wished grand success of today's event. And without their support and encouragement, this event would not be possible. So I'd like to thank all our three principals. And now, as we all know, that today marks the birthday of very important scientists. One is our legendary Indian physicist, Nobel laureate, Sir C.V. Raman, and the other one is, of course, Madam Mary Curie. And India celebrates National Cancer Awareness Day today because it coincides with Madam Curie's birthday. So now we'll have a brief tribute session to Madam Curie, and which will be done by Shuchandana Chaudhuri, semester 5 student of Department of Microbiology, Michael Madhusudan Memorial College. Shuchandana, the platform is yours.
Thank you, ma'am. I'm Shijandana Chaudhary. Good evening. I'm here to commemorate a lady and one of the greatest scientists on Mother Earth who devoted her life in research of the cancer treatment. Maria Salomia Sklodowska-Curie, known simply as Marie Curie, was a Polish and naturalist French physicist and chemist born in Warsaw on November 7, 1867. The daughter of a secondary school teacher, she received a general education in local schools and some scientific training from her father. In 1891, she went to Paris to continue her studies, where she obtained licensureships in physics and mathematical science. Her early researches, together with her husband, were often performed under difficult conditions. Laboratory arrangements were poor, and both had to undertake much teaching to earn a livelihood. In 1896, Henry Blackwell discovered that uranium salts emitted rays that resembled X-rays in their penetrating power, and these radiations did not depend on an external source of energy, but seemed to arise spontaneously from uranium itself. Influenced by this discovery, Madame Curie decided to look into uranium rays as a possible field of research. Using an electrometer, she discovered that uranium rays cause the air around the sample to conduct electricity. She hypothesized that the radiation was not the outcome of molecules but must come from the atom itself. Madame Curie also developed methods for the separation of radium from radioactive residues in sufficient quantities to allow for its characterization and careful study of its therapeutic properties. Her findings and discoveries had also contributed in the treatment of cancer. She discovered radium and polonium that was used as radiotherapy and was first used in brachytherapy. Cancers of the breast, prostate, and brains were among the first to be treated by this method. Her findings also enabled the development of nuclear medicine using radioactive isotopes for cancer diagnosis and treatment. She was awarded twice with the Nobel Prize. Firstly, she, along with her husband, was awarded for physics in 1903.
Your voice is not coming, Shuchandana.
Hello.
Shuchandana, your voice. Shuchandana, your voice is not. Yeah. Now it's fine. Okay, continue. Continue.
So, Madame Curie died on July 4, 1934, at the age of 66 from aplastic anemia, a blood disorder caused due to prolonged exposure to radioactive materials. Although she knew that radioactive materials can be harmful, still she didn't give up working on radioactive elements and continued with her discoveries. She devoted herself to the remedial work. Madame Curie thus paved the way in the development of modern oncology and radioactivity. Her name is now synonymous with cancer research and radiation oncology. Thank you.
Thank you, Shuchandana. Thank you so much for the lovely and heart-touching tribute. So now it's time to enter our main lecture session. And it is with great regret that I informing you that Shapna Mukherjee ma'am could not be here today because her son has suddenly taken ill. Shapna ma'am is the real inspiration behind the West Bengal unit of MBSI. She is currently our national secretary. But in spite of her worries and anxieties about her son, she has sent her good wishes to us so that we continue with the seminar and make it a success. So now I'll request Kirat sir to kindly introduce our resource person, Dr. Mrinal Kanti Ghosh.
Okay, before that, I think Dr. Malini Ghosh, Malini madam would represent some few words from his college.
Okay, so Malini madam.
Okay, good evening to one and all, and welcome you on behalf of all our 3 colleges and also from Microbiology Society India to be with us on this evening on a special day, I should say, where we have the opportunity, and in one word we can say we are blessed to have the chance to organize such a webinar on this special day to make an awareness amongst those who already know the thing and also those who do not or are pretty late aware about the disease and its outcome and how to get really be very conscious of the fact. So my words from our Department of Microbiology, Dhruvochand Haldar College, as an officiating as HOD, I convey our heartiest gratitude to and welcome Dr. Mrinal Kanti Ghosh, Chief Scientist and Head, Cancer Biology and Inflammatory Disorder, CSIR IICB, the True Campus, and he is also the professor, ACSR, New Delhi. So welcome you, sir, and we feel all together, our team members, that it is an excellent opportunity to have sir with us to deliver a very pertinent topic on a very special day. Thank you. So welcome you, and I would transfer to Kirat sir to continue the session.
Thank you so much, madam. So we are approaching towards the main attraction of today, and it's a very honor for me to introduce a scientist like Dr. Mrinal Kanti Ghosh. Sir is very close to me, I think, close to me. And it's a great pleasure to introduce our esteemed speaker for today, Dr. Mrinal Kanti Ghosh. Dr. Ghosh is a highly accomplished chief scientist at CSIR Indian Institute of Chemical Biology, IICB, and professor of Academy of Scientific and Innovative Research, ACSIR, with a PhD in biochemistry from University of Calcutta. He has dedicated his career in advancing our understanding in cancer biology and inflammatory disorder. After a prolonged postdoctoral research in abroad, Dr. Ghosh joined Indian Institute of Chemical Biology. Dr. Ghosh has an impressive portfolio of research with very high impact publications, books, focusing on critical areas such as signal transduction in cancer and the role of chromosomal instability in immune invasion and metastasis. His recent publication in prestigious journals highlight his innovative contribution to the field, including insight into the therapeutic opportunities in lung cancer and in the implications of carbon nanotubes, very striking field, the upcoming field in cancer diagnosis and treatment. It's very much narrow for me to introduce the scientific background, the scientific arena of Dr. Ghosh in this short time. And in addition to his research, Dr. Ghosh has received numerous awards, including the Bharat Vikas Award and recognition as a Fellow of National Academy of Science and Technology. He has also played a vital role in mentoring next generation scientists through his involvement in academic evaluations and as a reviewer of several high impact journals He has guided several PhD students, postdoctoral fellows, and the name of Ghosh Lab in West Bengal as well as in India and abroad is very much prestigious among the academic communities. He is the reviewer of several prestigious journals. Dr. Ghosh is the co-chairman of evaluation in ICMR grant, council member for West Bengal Academy of Science and Technology, and the chairperson of Management Council, CSR ISCB True Campus, and a senior scientist in charge in CSR ISCB True Campus, Salt Lake, Kolkata. Today, we are fortunate to have Dr. Ghosh share his insights and expertise with us on the day of National Cancer Awareness Day. Please join me in welcoming Dr. Mrinal Kanti Ghosh to the platform. Sir, please initiate your lecture on the platform. Thank you, sir.
Yeah, so I'm actually Deshmukh sir, Patankar sir, and all the organizers when invited me to give this kind of lecture on the special day. I'm glad to accept, and I want to deliver in such a way so that means I can enjoy with all of you in this evening. And I will focus that on the topic that you decided, and you tell the story about that things that the cancer awareness, that is the most critical issue of the society, that everybody should aware about this. So if hundreds of people join this lecture, they can actually convey the message to the others. So without wastage of time, so let me actually start the talk. Give me just a minute to open this window.
I'd like to request all participants that during the lecture, if you have any question, then you can type it in the chat box and we'll take them up during the interactive session.
Kirat sir, may I request you to ask? Okay, no, it's just there. Okay.
No, it's visible. Yeah. So your screen is visible.
You can push go. Hello, Kirat.
Yes, sir, it's visible and you are audible too.
Okay. So I'm clearly audible, right?
Yes, yes.
Okay, so let us enjoy
Full screen.
this
Is it full
evening.
screen?
Yeah, it's full screen.
It's full. So, okay, let us enjoy this evening with the integrating biology and emerging technology, a holistic approach to understand the cancer, the dangerous disease. And we know that victims and victims' family and the friends, they are worried when there is in a family, someone is the victim. And we all know that what happens at the end. And there is a possibility of cure the disease, but sometimes what happened, we are locked, we cannot do anything and we cannot cure the patient if the patient is already in later stage, like the stage 3, stage 4. And let me thanks to the organizers, Deshmukh Sir, Pathankar Sir, Kirat, and Malini, and the Raidegi coordinator. So they are organized this particular event, and they are actually selecting the topic on this special occasion. And I'm happy with that and happy to share with you my vision. And I hope that I will cover most of the things in the cancer research and treatment. And I will, of course, touch that in the microbiological things. But I think you all better than me regarding the microbiology. And let me introduce myself. I was born in a village, and then my entire education since that is the higher educations, BSc in chemistry honors, then MSc in biochemistry and PhD in biochemistry from University of Calcutta. Then I moved to USA and returned back to the origin, the same Bengal, where my academy started, where I am educated and where I have to serve, and I am servicing right now. So before going to my lecture, let me tell you that in cancer, there are 10 Nobel laureates. And this year, Victor Ambros and Gary Rubkun, they are actually awarded the Nobel Prize this year because of their discovery of microRNAs and their role in gene regulation. That is very important topic in particular this decade. And in the last one is 2018. That time also in the immunotherapy field, the Nobel laureate was Nobel laureate from that same cancer field. And here you can see that since 1926, this is the 10th Nobel Prize awarded to this particular area of research. And before going to discuss about the cancer, we have to know about our normal human body. What are the organs are actually helping us for everyday life? And these are the organs like the brain, heart, stomach, kidney, bladder, intestine, liver, lungs, thyroid, eyes, ears, pancreas, spleen, stomach, prostate, the male-specific, breast, ovary, cervix, woman-specific organs, all we need in a normal function. But the problem is that because of some kind of events, that is non-functional or over-functional. And that is the problem of this disease. And this disease can be caused by hereditary reason, even that epigenetic change, gene mutation, infection by the infectious agents, carcinogen, smoke, diet, evolution, all are claim that particular disease. And here you can see some pictures that of the microbes that can cause many diseases, including cancer. Not all cancer, but some specific cancer, when that particular infectious agent infect a cell in our body, and our body composed of billions of cells. And inside the cell, there are specific organelles that we all know, like the nucleus containing the hereditary material as a DNA, mitochondria, the powerhouse of the cell, endoplasmic reticulum, lysosome, ribosome, Golgi apparatus, all are helping different processes. But the major process is that the central dogma we know, DNA to RNA to protein, that in our body, all cells, this particular dogma is propagated. So there is replication, transcription, translation. And when one cell divides to two daughter cells, before that, they have to complete the entire DNA duplication. So that means DNA will be replicated and make all DNA content just a double amount. And that is not enough for the cell to divide or take a decision. But along with the replication process, there are the parallel processes are controlled, that is transcription, translation. And transcription, that is the message is actually stored in the DNA, in the chromosome, that is actually transcribed to the different forms. And one such form is called mRNA. And that is the messenger RNA. That means the message is inside that particular RNA. And that transcript then converted to translate, that means decoding. So that the message is decoded to another form that is polyamino acid chain, that is called polypeptide, but it's not a protein. So that means this is continuously working in each and every cell. And that polypeptide, slowly it is converting to that different type of modifications. It is going to be matured and producing a particular protein. And that protein is not enough for all functions. Protein may be the structural or the functional. But sometimes what happens, most of the proteins, they need some kind of modifications like acetylation, phosphorylations, methylations. It's not like that each and every polypeptide will be matured to protein and the proteins will be pushed transnationally. It is the conformations will gain, and that is all maybe the right conformations. So there is a possibility of non-conformational proteins that will be degraded or by the proteolysis process through ubiquitination. And there is a balance things is the ubiquitination to shape that particular. So that means that balance is going on. So that cell is maintained, each and every protein in a particular threshold, and then it can do normal function. And there is several processes that is control the proteins inside the cells. And the 20,000 genes are transcribed actively in our each and every cell. So 20,000 messages are actually decoded. There is a huge, and each and every one is under control by different organelles or the different proteins inside the cells are controlling the proteins. So protein control protein. So that means each and every protein have some kind of restricted function. And suppose that proteins, when it is active, they have a lot of different functions inside the cells for cell division, cell differentiation, and other processes. And proteins within the cell, they control the cell cycle, and we know that each cell is cycling, and that means it goes to G1S, G2M phases, and then it's divided and produce two daughter cells of the similar type. And this process is going on that when one egg cell is actually fertilized by the sperm, then it is producing two daughter cells of the same picture. And in our body, we know there are biological clocks and there are another invisible clocks. And that invisible clock is everybody's systems is there. And the clock ticks on, the intrinsic design limitations and trade-offs of our bodies eventually come into play. And that is called transformation. And in a cell can be transformed to another property. Either it will be die or it will live. So that means it will getting extra power or it will lose some power. So that different kind of things happens. And eventually what happens, the cancer is evolved. And there are two models, like the stochastic model and the deterministic model. And what happened, that evolution, the billion years of evolution evolved that something will change always. And there is a variation. And that variation, so when that is fit for the system, because of this fitness, there is a selection process started, and that cell is selected for getting or gaining another property. That property helps the cell for extra power. So evolution is helpless to eradicate anything which manifests post-reproductively. So you can tell in such a way that most cancers arise at the post-reproductive age of the lifespan, and it is evolutionarily neutral. And because of these kind of processes, evolutions, or the different effects that infection or epigenetic change or genetic change or something else, the cancer can appear, and there is a huge cancer burden worldwide. There are different organs that can be affected, and all the organs are not alike. So that means they have different cause for these cancer productions in a particular organ. And the major cancer types like the prostate, colon, lung, breast, and several others like the bladder, kidney, brain. And you know that statistical analysis that actually Pathankar sir and the Deshmukh sir, they are explaining that things and that calculations in the global perspective in 2030, the new cases will be 21.7 million and 1 million is 10 lakhs. So now the figure is what will be. And there will be a 13 million cancer deaths that is predicted in 2030. So that is a huge burden. So cancer awareness is very important. Cancer awareness is not today. Cancer awareness for the last few decades. The people are trying to avoid about the cancers to the people who don't know about the cancer. And the mortality rate, you can see, the lung is the huge, the highest mortality. Then stomach, liver, colorectal, breast, esophagus, cervical cancer, uterus cancer, pancreatic cancer, prostate cancer, bladder cancer, brain cancer, kidney cancer. The question is, what is the origin? And here you can see there is a highlight of the brain CNS. That means brain central nervous system. Why it is highlighted, but the mortality rate is not so high? Because the thing is, this is the vital organ in our body. That organ is actually controlling all other organs. Even our skin, that if there is some mosquito sits on the skin, then brain can tell where and when, how many are actually sits on our skin. So that means it is so vital and so sensitive organ. And the causes are, there is the genetic cause or the physical change, lifestyle, hormones like that, two hormone-dependent cancer like the breast cancer in female, prostate cancer in male, that is hormone-dependent. Breast cancer, ER alpha dependent, estrogen receptor alpha dependent, and prostate cancer is androgen hormone dependent. So origin maybe that if we think that in our body, almost more than 98% cells are differentiated cells. And some cells are stem cells, some are proliferating cells, the progenitor cells. And that is always maintain that balance. But there is a possibility of proliferating cells come back. That is overproductions. That means it is getting the overproliferation, lost the control of proliferation, or the differentiated cells who are exit from the cell cycle, they can come back to the cell cycle, and because of that de-differentiation, they can start proliferation. And if the system lost the control, then cells continually divide, and ultimately producing benign or malignant type of tumors. In 1997, after completion my PhD program, so I went to Cleveland Clinic Foundation, Cleveland, Ohio, USA. I worked there almost 8 years. And that time I thought that what I have to do, which area I have to start working. So I started working with the epigenetic control of the cancer. So that means the core area, that how the genes are epigenetically controlled, and started with a viral system, the adenovirus. Though how adenovirus infect and produce the oncogenic transformation. And this, each and every adenovirus, papillomavirus, AC40, large D antigen, like that kind of things, all these virus, they have early proteins, they have late proteins. And the early proteins E1A, E1B, E1A can dislodge the chromatin structure like that. It can target the P130, that the RB family protein. RB is the retinoblastoma protein. So it can dislodge the chromatin structure and turn on the gene expressions of the cell cycle genes. So that means something should be triggered. And that triggering effect by epigenetically or genetically or by any component of any infectious agent can trigger this phenomena, and it can be initiated the process of oncogenesis. Then after 8 years, relocated to India, came back to Bengal and joined CSR Indian Institute of Chemical Biology, 2007 to 2016. That time, I'm mostly focusing on prostate cancer, colon cancer, breast cancer. Why 3 different cancers? One in male specific, one is female specific, another one is the common thing. And I worked in USA in the brain cancer, but I cannot start at the beginning the brain cancer research in this particular platform. So I have to create the platform, then I have to start for that vital cancer. And mostly working on the signal transduction in cancer to identify the right target in cancer to develop the medicine against those particular target, and that is the India's vision. And after 2016, I moved to the current campus, the Salt Lake campus. Since 2016, I'm working here. So my group is working hard on these three and brain, so 4 cancers to understand the cancer biology. And in all these cancers, we are focusing on signaling crosstalks, so signal transduction pathway, how they are crosstalks with one with another to empower the cancer cells for the tumor development. These pathways like the EGFR pathway, NKI pathway, ER pathway, Wnt-beta pathway, and the gene regulation. So that means how genes are regulated, how they are overexpressed or underexpressed. Protein turnover, that means how the proteins are maintained, their threshold inside the cell. So some proteins are destroy other proteins. And also the delivery of the molecules, that is very important because we are taking medicines, but we don't know how much medicines are actually goes to the destination where the tumor is. And I will explain that part later, that peptides and small molecules based drugs, how we can deliver it. But our goal, we set at that time, 2007-8, that clinical investigations, that what the clinical questions, how we can address the clinical questions through the basic and applied research. That was our goal. And we tried to stay in that particular goal set. And we all know that 2011, Hanayan and Weinberg, they actually wrote a nice review on the hallmarks of cancer in the Cell Fresh. And that those hallmarks like the sustaining proliferative signaling, evading growth suppressors, so that growth suppressors cannot be prevent that growth process, activating invasion and metastasis, enabling replicative immortality, inducing angiogenesis, that is very much required to supply the oxygen and the food to the cancer cells, resisting cell death. So cell death should be resisted. So that means tumor can be developed. And this particular review, again, 2021, they are actually re-edited. And at the beginning, we started the clinical investigation like that. We are trying to get the human tumor tissue specimen through the human ethics approval from the clinic. We are taking 2 sets of samples. One set, we are doing the post-operative samples for gene expression pattern analysis and epigenetic regulations. Then other parties, we are working on that formalin fixed tissue sections for protein expression analysis and correlation studies. We always try to know that what is the gene expressions and how it is that correlated with the protein level. And protein as the ultimate molecules that is actually changing everything. So our candidate proteins, we're working on the RNA helicases, kinases, and the protein threshold maintenance, enzymes, families, E3 ligases, debucotinases, the receptors like EGFR, SSTR2, tumor suppressors, PML, p53, and PTEN. There are some few others like FOXO3A, FOXO family proteins, but there are many oncogenes. There are countable tumor suppressors, but uncountable oncogenes. Those are discovered and those not yet to discover. So we will enjoy now that about to know that what are the different major cancers and what happens, how we can diagnose those, how we can understand the cancer processes. In prostate cancers, what happened that generally the diagnosis can be done by the digital rectal exam. And after that, if the doctor is thinking that there is a possibility, then PSA test, the blood test. PSA is prostate specific antigen. And generally, this is the disease of the aged people that men, when it is crossed the 45, 50, every year have to maintain this test to understand that PSA is within the level. If cross the level, then further analysis may be required or treatment may be required by clinicians. And there is another one that is called the PSCA, prostate stem cell antigen, a novel prognostic marker in prostate cancer. And this is under clinical trials. And for metastatic castration-resistant prostate cancers, this cancer remains lethal. There is no drugs are available. Recently, one chimeric antigen receptor CAR T cell therapy is actually in the clinical trial. So if it is coming to the market, it will help us very nicely. And the clinical trial, the post-cells are the directed T cells in men with that metastatic castration-resistant post-cancer treatment. Here in the picture, you can see there is two picture. One is normal prostate, another is the prostate cancer. And you can see that you can clearly visualize that what happens when it is in cancerous stage. The architecture is totally lost. And in the right panel, you can see that hyperplasia versus neoplasia. So Ki67 is the marker for the proliferation. So that means it is huge in the neoplasia. So that means neoplastic tissues that is highly proliferative. And if you check that the other cell mass of these two left and right panel, you can see the cell number increases. And there are multiple protein factors that are changing. So as per our requirement, we can analyze that what particular diagnosis we need for which particular protein status we have to check. And out of 6 men, 1 man may be targeted by this particular disease. And that treatment depends on the different stages, like the T1, T2, T3, T4, the 4 different stages. And this is a slow progressing disease compared to any other cancers. So we can do the watchful waiting and perfect treatment procedures. There are procedures like the radiation therapy, hormone therapy, vaccine, surgery, chemotherapy, cryotherapy. Vaccines are available. Hormone therapy, there is at the beginning, doctors are actually prescribed for the androgen ablation therapy. So if you remove androgen, what is the response? Then the second level of treatment. And surgery, chemotherapy, and radiation therapy is common to all cancers, but not the same radiation therapy, not the same dose of radiations. It is specified by specific cancers. Let us know about the breast cancers. In the breast cancers, we know that there is a particular subtype called the TNBC, triple negative breast cancer, that is dangerous. Breast cancer is self-dangerous, and triple negative breast cancer, that it is hard to get the patients in the cure form. And there are some genes are responsible like the BRCA1, BRCA2, and hope you know, couple of years back, there is a story about the BRCA1, BRCA2. And there is another gene is HER2, and the p53 tumor suppressor. That p53, when it is wild type, it is tumor suppressor, but when it is mutated, specific mutation, making it highly cancerous phenotype. So that means that p53 is dangerous when it is mutated. And the disease process like normal to hyperplasia or dysplasia, then in situ carcinoma, then adenocarcinoma, or that means you can metastatic carcinoma. And when we carefully looked about the gene clusters, we observed that in the panel, middle panel, you can see there is a transcriptome analysis where you can see E-cadherin, FOXO3A, PML, these are actually reduced. That is, the green is high, red is low, so they are reduced, whereas N-cadherin, survivin, vimentin, here alpha, they are overexpressed. So that means these are responsible for the metastatic progression, and that cells can be metastasized. We can also do the different other factors we can check. We can do the validation of different results of the in vitro results. We can do by tumor development, that is by xenograft modeling, or we can use the nude mice, and we can check that what are the genes responsible for that oversized tumor development. And out of 8, 1 woman can be affected by this particular disease. And there are different stages like that. Tumor can be developed in the glandular tissues, and it then metastasizes to the lymph vessel. It can go to the other tissues, and that is happening in other cancers also. And it can grow in other organ. So one organ, when affected by cancer, then another organ can be affected by the same disease. And there are treatment procedures like local therapy and systemic therapy like chemotherapy, hormonal therapy, targeted therapy. So more drugs, more effective drugs are required, more effective targeting is required. What about colon cancer? That is because of the lifestyle disease. Deshmukh sir and Patankar sir is telling that, oh, there is a problem in the lifestyle. Yeah, that is very much important about lifestyle. And generally, when colon area there is a pain, then we go to the doctor, and doctor is generally prescribed that the colonoscopy. And colonoscopy is actually can tell you that if there is the abnormal growth in any position. And if that is happening, then we have to do further test, and we have to do the ISC or biopsy analysis of that particular few samples. And out of 20, there is one person can be affected by colon cancers. And there are 5 stages, 1, 2, 3, 4, 5. At the 5th stage, it is spread to the other organs. Here in our lab, we did a lot of works on colon cancer in last 10, 15 years. And all the data we validated by the mice model. So we actually implanted the cancer cells in the flank region, and then we developed the tumor. And then when we treat with the different compounds, we check that it is increasing or decreasing some genes like the P68 is RNA helicase. When it is overexpressed in particular cell, then it is stably expressing and producing bigger tumor compared to the original cancer activity of this particular cell. And it can be metastasized, so it can go to the lung, and lung nodules can form. And we observe that when P68 overexpressed, it is producing more nodules, more number of nodules, and the bigger size of nodules. So that means P68 is oncogenic. Like that, many other genes, we explained that why that is oncogenic, how we validate in the animal model systems. Regarding all these cancers, the prostate cancer, breast cancer, colon cancer, there are diagnostic tests that are available, like lung cancer, CT scan, and there are some markers like the EGFR, ALK. These genes you have to monitor. Not all the 20,000 genes you have to monitor. We have to monitor for specific genes. Those are marker for particular disease. Like prostate cancer, PSA, that I explained before. In breast cancers, generally mammogram, MRI, that is important. And we have to investigate about the status of the BRCA1, BRCA2. The BRCA1, BRCA2 mutations or overexpressions or anything. In colorectal cancers, colonoscopy, but most important genes are KRAS, NRAS. So we have to investigate about the status of the KRAS, NRAS. Ovarian cancer, again BRCA1, BRCA2, and ultrasound can be the preliminary stage, primary test. And the leukemia, generally blood test, CBC, and the bone marrow, the biopsy is important. So these are few examples, but each and every cancer, there is particular test, there are common tests. Common test for multiple cancer, but not all cancers. After that, we decided that for the cancer research, we need huge amount of money and we need huge amount of knowledge. So that means inside a cell, if we do not think about the glucose, about the metabolism, about the DNA, about the RNA, 20,000 proteins are there. So now you can see how complicated the system is. Now we have to decide how we can carefully look after those things, what is required. But it is systematic events. That means proteins are functioning, they are systematically, roadmaps are there. There are some pathways are there. There are some proteins, they are organized for a particular functions. Proteins are localized in a particular area. That means lysosomal protein, the proteolytic enzymes, those that go to the lysosome. The powerhouse, they have separate set of proteins like that. So that means each and every protein, they are under control. So the complex systems, if we try to understand, we need huge amount of money, huge amount of knowledge. So collaboration is required. So we collaborated with the Mayo Clinic USA. We collaborated with the Systems Biology Institute, Japan. And that collaboration was 3 to 5 years in last decade. Now, tell you after this collaborations or even that I started this work when I was in USA in the brain cancer research. But let me tell you that, let us know that what is the beauty of this particular disease? Why so critical? Why it is highlighted to the worldwide society? A tumors in the brain that can be originated is a de novo, that is directly at the grade 4, or progressive, that means grade 2 to grade 3 to grade 4. It is converting with more and more mutations. So that means there is a driver mutation. After the driver mutations happening, the cells getting some change abnormality, and then cells dividing, and during divisions, they gathered the more and more mutations of the sensitive oncogenes. So that means that progression of the disease process from stage 1, 2, 3, like that. But the grade 1 is generally non-malignant type. That is pilocytic astrocytoma. That is happening in case of child. Even that less than age of 15, 20. And above 20, mostly are astrocytoma, anaplasma, glioma. But there are other brain tumors like meningioma, pineal region tumors, medulloblastoma, cerebroastrocytoma, brainstem glioma, pituitary tumors. There are several others. This not only the countable things. Here you can see exactly 120 types of brain tumors till day it is reported. All are treatable, but some are not curable. The treatment options like the steroid therapy, like surgery, chemotherapy, radiation therapy, stereotactic radiosurgery, and even nowadays the gamma knife. I will talk about the gamma knife also. And there are some particular brain tumors like glioblastoma, anaplastic astrocytoma, and low-grade diffuse astrocytoma that we are mostly working, and altogether these are called the glioma. The question is glioma. So dangerous. So how we proceed? There are some symptoms will be there, so that means some normal people can be think about that something is there, some problems in their brain. So they have to go to the doctors. They have to examine. There are symptoms like the sudden headaches, difficulty with balance, dizziness, nausea, vomiting, vision impairment, hearing impairment, seizures, like that kind of symptoms can appear. And that doesn't mean that there is a brain tumor. Something can happen not as a tumorigenic system. And there are causes like the age is the common factor, and common in children and the older adults. And that was in the 1 decade before, but nowadays, no, not the children and the older adults. Any age can be affected by the brain tumor. Gender men are more likely than women. There are infectious agents like viruses and allergens, some cause that can goes there, and that can happen. Electromagnetic fields, if we always be affected by that, that can cause brain cancer. Like you know that always that telephonic systems are always telling that mobile can affect this. I'm not sure. Ionizing radiations, obviously. That means previous some treatments, the ionizing radiations are affected something as a result of that cancer can happen. Some head injury and seizures sometimes happens, but after a couple of years, after a decade, that brain cancer can appear. And there are different grades. You can see that the cells are packed, more and more packed, and ultimately they are metastasized. But this is the only cancer that secondary tumor can happen, but secondary tumor within the brain, because brain cell cannot go out from the brain. There are some barriers. So that means sometimes what happens that other cancer cells can go in the brain, but brain cells cannot go. And there are diagnosis and treatments are available. The major treatment procedures of cancer include the surgery, radiation therapy, and chemotherapy for all cancer. But in case of the brain cancer, there are some specific. Radio imaging, that is like PET scan, CT scan, MRI, SPECT, ultrasound. Every applications have some specialty and they have some special applications. So that doesn't mean that anything can be implanted for particular process. So anything cannot be used. So that means proper investigation by the clinicians is very important. In brain cancer, you will know that MRI is very important. And nowadays, the PET and PET CT scans are there for several cancer types. In our lab, almost 10 years back, we also worked with the radiomedicine. For diagnosis purpose, we actually took that one particular peptide we developed, and that peptide we tagged with a radioactive material called technetium. So that is the radiomedicine. That radiomedicines we worked thoroughly in the in vitro systems in our lab. Then we applied to the tumor systems in vivo. So how? We developed the tumor intracranially inside the brain. So when the tumor is developed, after that, what we did, we compared that with injecting the radiomedicine through the tail vein. It goes to the blood and throughout the body. But you can see the top picture, the brain image is there within 15 minutes, and after 30 minutes, that is clear. Nothing is there. And in both the cases, you can see ultimately bladder is highlighted. So that means radiomedicine goes to the bladder, to the urine, and it goes out from the body. So body cannot be affected. So that was reported in 2012. And treatment of brain cancer is very important, and there are continuous development of the brain cancer treatment. There is the contrast enhanced MRI. There is a craniotomy with 5-ALA fluorescence dye, so that can be fluoresced and giving the different kind of color to the tissue versus the tumor tissue. External beam radiation therapy. And there are a couple of drugs are available that is applied for the brain cancer patient. One such drug is temozolomide, or it is another name is temojam or TMZ. And that drug is huge costly. That is chemo. That when brain cancer happens, then all the time surgery cannot be done. Because of that, the position of the tumor. In some positions, if the clinician, the surgeon, do the surgery, then there is a paralysis can happen in the body. So all the brain tumor cannot be removed by surgically. There is a treatment procedure, the radiation therapy. There is a treatment procedure, the chemotherapy by temozolomide. And temozolomide, like 100 milligram drug, is cost around 13,000. And 250 gram around 21,000. Huge, costly. Every day you have to take one capsule. That is problematic. And during radiations, generally 100 milligram capsules are taken by the patient, and there is a 6 or 7 cycles, total 35 radiation cycle. After that, there is a gap of 6 months to watch the patient. But in what is the record is that in last 20 years of research and clinical and the basic research, that is giving only 3 months of lifetime, additional lifetime. So that means grade 4 brain tumor patients generally dying within a year, within 12 months. After the different several treatments, even the gamma knife, it is extending 3 months, that means 12 months or 15 months. So that means if the brain tumor is grade 4, then lifetime is 15 months after all treatments, all costly treatments. And if it is a lower grade, then maybe 15 years for the grade 2 and 10 years, 5 to 10 years for grade 3. So that means early detection is very important to provide enough time of the patient as a lifetime, for the lifetime. And here you can see that some images like that, the brain images and brain surgery and radiation surgery, how the radiation surgery can happen. And there are other techniques like the brachytherapy, the radiotherapy like the gamma knife. A gamma knife is nothing but the gamma rays are actually coming from the beam of gamma rays coming from several directions, 360 degree directions. So all the gamma rays are actually pointing in a single point. So that means all the gamma rays are actually accumulated in a single point where the tumor is. So the idea is to burn out that particular area. So that means the tumor area, we are just burning out. So cells will die. So cancer, the tumor will be sink, so reduced. But after that, we did the treatment of the chemotherapy. And there is another problem is that the cancer stem cells, we all heard about this. So those stem cells, not the normal, because in the normal tissue, they are normal stem cells, but these cancer stem cells, they always involved for the recurrence. That means tumor is shrinkage. After that, after treatment, if the tumor is resistance against the chemotherapy of temozolomide, it can reappear again. But many times surgery is not possible. We are working in our lab for the last 2 decades regarding that how we can understand, how we can develop, or how we can find critical targets in brain cancer research. And after the target identifications, we always try to validate the target by not only the cell culture model, cell culture systems, but also in the animal model systems. And we generally use the rat as the animal for this particular purpose. And rat C6 glioma cells, that is mimicking the human glioma cells, we are implanting after the removing the skull, and then we implanted nicely in the frontoparietal lobe of the brain. And then we wait. Within 10 days, we understand that the tumor is developed because something problem in the rat. So rat is some dizziness appears, not taking the proper food, amount of food or the water. And so that means within 18-20 days, we see that the rats are dead. But if we started treatment after 10 days of implantation, you can see this figure. Here you can see the figure B. Figure A is actually that it is a kind of in vitro BBB crossing technique. So we can check that drug can cross the blood-brain barrier or not. So we actually use the insert that is actually one side is tightly packed endothelial cells, the other side is astrocytes tightly packed. It cannot easily, that any molecule cannot easily pass through the systems. But when we use the nano formulations, that nano formulated molecules, they are actually passed through the systems that we can use by imaging techniques. And that particular nano formulations, when it is TAMRA tagged, that means nano formulation on the surface, there is a tagging of against a receptor that is specifically overexpressed in the cancer cells, brain cancer cells. We design a peptide, and that peptide is tagging with a fluorophore called TAMRA. That peptide tagged, fluorophore peptide tagged nanomedicine, when we inject it through the tail vein of the tumor-bearing rats, you can see nice image, red image there, where the tumor is, where we developed the tumor. We implanted the cells, but in the normal, where we didn't implant it, it cannot be highlighted. So that means it is not, drug is not accumulated anywhere in the brain. And we all know it is slowly out from the body through the urine. And we all know that in each and every cancer, the single drug-based therapy is not giving good outcome. Like the platinum group compound, like the cisplatin, carboplatin, oxyplatin, they are generally used for the breast cancer treatment. But sometimes, we need the combination treatment, like that particular drug treatment along with immunotherapy or along with radiation therapy. So that means combination therapy is important. We are working for combination therapy, like the peptide-based and the small molecules that I think at the beginning in the slide, I told you that I will come back to that point also. And I explained that there are 3 important tumor suppressors like the RB, p53, p10, and the FOX family, of course. We worked very important issues of the p10, the stability of p10, why p10 is get lost in tumor. And generally what happens is the chromosome number 10, and p10 is actually named from there, and chromosome number 10, it is lost. But sometimes what happens in most of the patients that p10 is there, but truncated version. So we worked on that truncation, why it is truncation, and how we can keep it stabilized inside the cell so it can prevent the disease process. So we developed that peptide, p10CT. It's a 48 amino acid containing peptide. We packaged in exosome. I'll come back to that, what is exosomes, how we package it. And we also package the EGFR inhibitor because EGFR is a receptor and it is actually overexpressed in most of the cancers. In brain cancer also it is overexpressed. So we are using that DIM, diinterlyl methane, that particular natural product-based molecules we actually packaged in a nanoformulations that is prepared with the PLG. And we tagged a peptide, SSTR2 peptide, because somatostatin receptor 2 is overexpressed in glioma cells. So that receptor, for targeting that receptor, we developed that somatostatin peptide, and that peptide is actually tagging with a molecule, that fluorophore. And that two molecules can be used together to treat particular brain tumor model in rat. So that means it can give some better effect. And also we are working with the brain tumor samples we are actually receiving from the Park Clinic, Kolkata. My collaborator, Dr. Sandeep Chatterjee and Dr. Uttara Chatterjee. Sandeep Chatterjee is a surgeon, neurosurgeon, and the Uttara Chatterjee is actually IPGMR Kolkata. She is the pathologist. We worked together for long, almost 15 years. And we are getting the samples, and those samples we are doing the transcriptome analysis to understand the disease process, to understand the cancer pathways, or cell cycle regulations, or apoptosis regulation factors, or puberty signaling or wind signaling, which one is disturbed and which one is aberrantly activated for this particular disease. So that means our target will be there. So combination therapy, we can target, along with temozolomide, we can target that particular. And temozolomide is a compound that is actually damaging the cells by DNA intercalation. So it is actually modifying the residue, one residue, and that is methylated, that residue. And there is a protein called MGMT that is methylase, that removing that. So ultimately getting the chemo resistance. I am not going in deep in that particular area. So now, better to enjoy this evening, so we have to go a little bit in the innovative technologies and cancer treatment. And we all know that CRISPR, robotic surgery, cryo microscopy, although along with those, the artificial intelligence and the telehealth that help us a lot. And there are many techniques are actually under development. Here you can see in the picture of robotic surgery. So now what happens? Previously, doctors manually make all surgery, either ovarian or breast or brain or prostate. Everything should be the manual. So open and then do the surgery. And then it was recently what happens that they are using some instrument. So that means they are producing 3 holes and make the surgery. And now the techniques is robotic surgery. The total things is by a robot, it can do everything to remove properly from the region. So there is no manual error, but maybe the instrumental errors should be there. So we can be achieved one day that instrumental error is totally gone. So that means each and every surgery will be very nicely, it will be done, and it can remove the entire tumor mass from the tissue, from the organ. And there are some medical breakthroughs like that 2020, there is the COVID-19, and we all sucked about that. And we struggled for the development of the vaccine against the disease. And ultimately, many countries developed the vaccine, and vaccine saved huge number of lives. Although we sacrificed many, but we achieved. And now we don't have any fear about that particular COVID-19 disease. We know that things, if we fight, we can win. And there are some other diseases like the cervical cancer. There is a problem that is human papillomavirus caused the disease. And for the disease, women actually have to go for pap test. And you know, there are vaccines that are available, and that is called the Cervarix. Initially, doctor tried to prescribe the vaccines, but people do not take that vaccine because there are some problems appears at the beginning. But after 13th, I think each and everyone should take the vaccines if the health is okay. There is another innovative lung cancer vaccine, is BNT116, that is mRNA-based vaccine. That is also in clinical trial. Hope soon it will be in the market. there are vaccine types like the hepatitis B vaccine, the Sipuleucel-T, that is for prostate cancer, and there are other researches going on against the melanoma, lung, breast, and other cancer vaccines. And there is another popularity is coming that sometimes people are telling that personalized medicines, because we all look different. We all have different power. We all have different mind. We have different settings. And that is because of our hereditary, because of our DNA systems, because of that 20,000 gene expressions. So we all different because of 20,000 gene expressions. Some are in different level or different context. And the different gene expressions pattern can tell that what is the problem, what can be done. And here is the sample I am giving that the brain cancer is the gene expression of analysis of the GBM samples done from the NIBMG, National Institute of Biomedical Genomics. That institute, actually, they are doing that things, and then we analyzed it, and we observed that several of genes are actually changing. So one day, maybe the genome medicine will be available, and of course, it will be huge cost. I think only a % of people can achieve that things. Otherwise, it cannot be. Another most important issue that I can tell that chemotoxicity, because of that, when we deliver a drug, because chemotoxicity appears because of the chemical compound we are using for anti-cancer activity, and we remove the cancer burden. But the thing is that when we injected or orally taken the medicine, what happens? It can go to each and every cell to our body. So each and every cell can be affected because of that particular molecule. So chemotoxicity can appear. So the right way to reduce the chemotoxicity, we have to do something that can prevent the connection between each and every cell from the drug. So drug can be encapsulated by exosome, by nanoparticles. Another issue is that drug during delivery, like the peptide drugs, that can be damaged during circulations. So new technology is required for delivery of drugs to the destination where the tumor is. And especially for brain cancer, there is another problem, that is blood-brain barrier. And this is the picture you can see that different type of cells are engaged for that, this barrier. And this barrier is not a real barrier. It's a tight junction. And through the tight junctions, nothing can easily pass. And there are the agents like alkylating agents, platinum complexes, antimetabolites, cytotoxic antibiotics, spindle poisons, even the topoisomerase inhibitors that can be used for the cancer treatment because they have the anti-cancer activity. So novel technology that we are pursuing last 10 years, 12 years, that is nanotherapy. That means we package the toxic molecule inside the liposome or PLGA. Mostly we are doing the PLGA. And there is exosome. I think 2015 we published that particular research paper. The peptide therapy, that pTNCD can be packaged. So natural nanovesicles. So that means each and every cell always secretes vesicles. And those vesicles are called the microvesicles. One such microvesicle is the exosome. And here in the right side panel picture, you can see that exosome can carry mRNA, miRNA, several proteins, even actin. And these molecules can be, these vesicles can be fused with another cells and can load the materials to that cell. So that means cell-cell communication can be used. So if we can send this particular exosome-packed our interstitial molecules, and there is a target peptide tagging with that exosome, it can carry the load or the drug or the anti-cancer molecules to the cancer cells, and it is right target, it deliver it, and may help our treatment procedure easier. So in cancer treatment, so combination of surgery and chemotherapy or combination of surgery with radiation is very important. Sometimes only surgery cannot achieve our goal. So for the reasons we are doing this after surgery chemo or during surgery chemo, or after surgery radiations or radiations before surgery radiations. So combination is required. And recent new development is immunotherapy, targeted therapy, and the hormone therapy is not the new, it's old, but continuously it is developed. And the radiomedicines, radiopharmaceuticals, that is specific to cancer cells. Like example, AOH 1996, a small molecule PCNA inhibitor is a radiomedicine. It can be used, it is using actually. And there is another thing, there are, the organs are, why telling that the organs architecture is different, organ function is different, but inside the cells, there are many similarities, many dissimilarities. Because the 20,000 genes are expressed in almost all cells. So there are some similarities. So why not we can think about the repurposing? Suppose there are common things like that the platinum compounds or even the, you can tell the doxorubicin, that is also intercalating agents of DNA so that it can goes to cells and intercalate within the DNA duplex. So that means it can be used for multiple type of cancers. Like that, many drug molecules are available. Millions of molecules are actually from research it came, and there are many are actually in the market, and there are millions of molecules currently under research, maybe in the basic stage or in the clinical trial stage. So that means one day many drugs can come to the market. So that means now people are thinking about the repurposing. That means one cancer drug can be used for other cancers. We are also doing the repurposing that some colorectal cancer drugs, suppose we can test for the breast cancer if the target is right target. Like that, if we can think very easily that EGFR, it is overexpressed in many cancers. So EGFR specific drug can be utilized as a combination therapy of multiple cancer types. And this is the current therapeutic position in our lab. Not current. These are this is not up to date. This is I think before COVID. That is the position of the breast cancer research in our lab, glioma therapy, and the colorectal cancer therapy. And we actually mostly doing the basic science research. So identify target, validate target, cancer target, so that chemists can develop the molecules against those targets. And we got funding from several funding agencies like DST, ACRB, DST Nanomission, DST and DBT, ICMR, and CSR. We are also getting the CSR fund like that institutional fund, as well as that we individually apply to the DGCSR. So directly DGCSR grants we received. We also write the international grants to get some collaboration and collaborative effort. We can achieve something. Last but not the least, we all know mitochondria is the major organelles inside the cell, and that is the powerhouse. And we all know when we are in the school. And this mitochondria giving the power for the cell for anything, for any function. And what happened, you know? Like the plant systems, there are plasmodesmata. Through there, that one plant cell can communicate with another plant cell. So that means they are exchange their materials from one cell to another cell. In animal systems also, in human cells, they can produce the nanotunneling. So that means cells you cannot visualize. You need them high-powered microscope to see a cell. Now you can think about the research going in their directions that the nanotunnel among the cells that can be also visualized by using the high-powered microscope. And here in the below picture, you can see that is the pictures that how cells are actually communicating one cell with another cell. And in the reality, what happens? In the tumor, the cancer cells are having more mitochondria compared to the surrounding tissue cells as well as the immune cells. What happened? Why that disbalance? Because of this nanotunneling, cancer cells, they have high power. They are actually forming nanotunnel with the surrounding normal or the immune cells, and they are hijacking mitochondria from the immune cells and getting the power. And as a result, there is immune evasion in tumor. And that is one of the important hallmarks in cancer. And recently, we also wrote one review that in BBA Reviews on Cancer, that is USP7 that we are working as a dubicutinase last 15 years. That has multiple hallmarks that is actually working, that functioning by this particular molecule. So our vision is bench to bedside. So most important applications that we are till date we are achieving that exosome based and nanotherapy based treatment strategy of cancer. And that is we reached here because of because of my students. And before acknowledging my students, let me show you that Uttara Chatterjee, the pathologist, IPGMR, and Sandeep Chatterjee, that late Sandeep Chatterjee, because this year he expired. So this is Park Clinic, Kolkata, the neurosurgeon, and Suresh Bajoria, the urologist. They are helping a lot to this particular lab to get some knowledge and get some applications. And they're providing the tissue samples, the cancer tissue samples, as well as the adjacent normal areas. In this picture, you can see that the core 9 students is the first batch of my students. They developed everything. The Kiran, then Tapushi, Indronil, Shimana, then Anirban, then Orijit, then Firoz, then Momita. They are in the abroad. Many are abroad, like Kiran at Canada, Indronil, USA, MIT, Harvard, Shimana, she was in MD Anderson Cancer Center. Now Orijit and that Anirban, they returned from USA and now they are doing research here. Firoz in Glasgow, Momita, I think in the London. Binita, side by Binita is in the UCSD. And there are you can see if you go to the surface, then one by one added in that particular area of research. So there are research area that we are doing, the signaling crosstalks. We are doing chief is the E3 ligase. So here all 4 are working here. That Indronil, Satodipa, Shabusti, and Orin, and that in the other directions you can see Firoz, Satomita, Shani is currently working, Sauni is currently working in that group. Then is USP7, the house P, that is most important factor. That also started with Simana, MD Anderson right now, and then Gaurango, then Bhaskar, then currently Srija is working on that particular area. And the CK2 story that Anirban started that work, then Nilanjana worked, and then Nirajana worked in that area, currently working by Shubhojit and Mowli. And the drug delivery, drug systems working, the Orijit and Nilanjana joined that particular group. And then 2 women scientists, actually, one is Mita is currently doing as a woman scientist, and Sivani is completed the woman scientist and again applied. There is another woman scientist, Savannah, is woman scientist Wise PhD. She is actually got the Wise Kiran PhD program fellowship. And here is Afrin. She worked 1 year currently in Australia for PhD. Nilanjana again returned back to the lab and as a woman scientist. And that's all for my students. Now, for you all, a take-home message. Take-home message is every tumor is different because every people is different. Every tumor is different. So every cancer patient is different. So we need personalized medicine, but low cost. We have to achieve that goal. Maybe that AI or ML, artificial intelligence and machine learning, that will help us to achieve that goal, the personalized medicine. And so our roadmap is target identification, target validations, and preclinical development, that stage. And we all know that prevention is always better than cure. So at least we have to do something for this particular disease. So we have to restrict the disease. We have to restrict the number of patients. The humans have been investing in cancer research for decades, only while evolution has been tuning cancer suppression mechanisms for over 1 billion years. So for that reasons, you know That clock is for everybody, but sometimes clock cannot trigger, but someone can trigger. But when it trigger, we don't know. So the high time has come when we must step back, and try to learn from the expert rather beating up the buses with nothing but futile outcomes. So we have to ask an expert, a clinician. We should not tolerate that the pain. We have to do the early detection, and that can save us. And last one message I can give you, that I think in October 17, so I actually presented in the Doordarshan, that is the Prasar Bharati. I in a meeting with Krishna Chattopadhyay, who was the Sanchalak actually. So that lecture is available in the YouTube. I think if anybody interested can view that YouTube. Thank you very much. So early detection saves lives. So now you can see here are the different ribbons of different colors for different cancers. This is not all, but there are some. This is some, but there are more. So cancer awareness is very important. So I will try this awareness program already. I actually submitted one grant to the funding agency for the awareness program, but not yet funded yet. But we don't need the fund. We have to be aware about that cancer particular disease and the disease outcome. So we have to spread this kind of knowledge to the common people who doesn't know anything. Thank you very much to all of you.
Thank you, sir. That was extremely informative and in such a lucid way. I think our students also could relate. And so thank you very, very much. And there are some questions in the, we'll go to the interactive session. So the first question is, how effective is the crosstalk between researchers and clinicians at state or national level so that the outcome can benefit the human population? I think it's from Kirat sir, right? I think Kirat sir wrote this question. Yeah.
Oh, it is me who wrote the question.
Oh, okay, Malinidhi.
Yeah.
Okay,
Anyway,
it's from
so
Malinidhi. Okay.
crosstalk, this is terminology. Actually, crosstalk is not in that particular research field. Crosstalk is everywhere. You know, in the family, in the school, in the college, in the university, everywhere. Crosstalk is very important. So that means knowledge sharing. So if we do not crosstalk, there is no knowledge sharing. And you know that knowledge is actually power. So when we do crosstalks, we gather knowledge. So our cell, inside the cell, what happened? Different paths are there. And that each and every path there is a power. So when that path, one path is crossed with another one, they are getting more power. Like that, the EGFR signaling when it goes to three downstream paths. Like one is called RAS, RAP, RAC, MKK, MAPK, and then goes to the nucleus. Like that, another signaling that is called JAN pathway, stress pathway. And that stress pathway is sometimes crossed up with EGFR pathway. So that means cells get more power. So that crosstalk is there. So in research, the collaboration is the crosstalk. So that means when we are collaborating with someone, we are exchanging our knowledge. That means that one particular researcher, one scientist, when directly involved with the clinicians, they are discussing the matter that what happens in the patient and what research tells. So that means we can address the clinical issues. So we can do our research at the clinical route. So for that reasons, it will be better to reach that what doctors required, what doctors need. Doctor cannot do anything if the research cannot tell anything. So that means first thing is discovered by the researcher, that is established in the systems, then clinicians getting that kind of information, and clinical research developed those instrumentations or you can see the treatment procedures, so that can be used by a doctor. So doctors should read the articles. I think the recent doctors, clinicians, every day they are reading the different articles that is coming from from, I think that at least New England Journal of Medicine, that is explaining many important issues.
I have a question. May I ask
Thank you.
directly, sir?
Yeah, sure.
Oh, okay. Sir, I have a question. Since we are on a platform, this is Microbiology Society of India, and this platform and today's online platform has more than 80% of students. They are from different levels, UG, PG, or PhD scholars. So my question is, what is your view that how to promote research? Because these are research-oriented subjects from the very beginning, from very UG level to PG level, keeping in mind the situations of our colleges. I can say in state or in all over India. So how can we promote the research concept and research mentality to the students? This is a little bit different from the today's topic, but when we have got you in such a platform, I think it's time to ask.
Okay, I have to take some water because this is a very good
sure.
question.
Sure.
Okay, so the answer is, we learn from the schools. We never learn from the college or university. If we do not go to school, we cannot go to the college or university. If we do not have to write the words, we cannot write sentence. So each and everything is important. And this is not right that the best school will produce best scientists. No, there are not the good school, but producing the best scientists. And this is not like that the best cities can produce the best doctors or best scientists. Then Nobel Prize cannot go to the other countries. Otherwise, it will go everything to the America, right? No, not like that. So that means each and everything is important. Most important issue is the individual. So that means who want to learn, who want to learn. The student, because one day you are student, one day me also student, everybody is student, right? So that means who is learning, that is very important. That means that whatever you are telling, whatever you are teaching, your memory is accepting and your memory thinking. So that means thought process is very important rather than the teaching process. The teachers always teaches in a good quality. Teachers always guide. The teachers are guide. But the thing that what you want to be, that is within you, within your memory. So you have to think about yourself. And nowadays, that internet facility, that media, that is helping a lot. And as a result, what happened, you know, sometimes we are claiming that students are not coming to the college, not taking classes, like that kind of things. We forget our time, because our time we always go to the college, university, because we need teacher, we need guide, because we have only two options. One is book, another one is the teacher, no one else. But recent days, you cannot compare with those days. Recent days, if anybody want to learn at home, he or she can learn a lot. Like that. But the thing, whatever the resource you have to give good quality of teaching, so you will be a good teacher. And we all try hard to be a good teacher, but someone is a good teacher. You got it? But whoever is struggling will be a good teacher.
Thank you so much. Really, it's thought-provoking. These are the points we should address when we are going to classes or something. Yeah.
May I say something?
Yes,
Sure, sir. Sure,
Yes,
sir.
sir, of course.
Yeah. The question of Kirat is very, very correct one. And this is the need of time. Actually, the research aptitude, it must be developed. As Sir said, Mrinal Kanti said, it develops at the school level. The schooling is very, very important. And there, the importance is for developing the learning skills. That is very, very important in the student. And another thing is that the questioning skills, they are also to be inculcated in the student. I feel these two skills, they are somewhat lacking in the student. and that's why we face some time a problem. So that is my observation through all these years which I put in my service for nearly about 40 years. Thank you.
Yeah, even I can tell one more thing, that some knowledge when we are studying that we achieve during our masters. But nowadays, those particular areas is actually taught in the school levels, in the 11, 12. 11, 12 students, they know the transcription, replications, translation, like that, all these things. Any more questions?
Yes, sir. There's a few more questions actually. Like this one is also from Malindi, I guess. What is the earliest symptoms for brain tumor that can be observed with respect to stage of that cancer?
I actually explained that things: headaches, dizziness, the hearing problem. Those things can appear, but it is again that tumor to tumor it developed. So that means there are 120 different type of tumors. The tumor position, which nerve is there, everything actually depends on that. So for the disease, there is no common symptoms. At least the dizziness, at least the pain, something, at least sometimes some seizures. That can happen. That is the early detection. And we cannot see anything, no? So for the reasons, we have to go for testing. And that is the first foremost test is the MRI.
So that means too early stage, it is hard to detect, right? Because that develops, and like the other, we can externally see it. And here, like for others, there are different blood tests and things like that. But for this brain tumors, that is most of the time it is more advanced stage, right?
Yes, you can tell one way that it can be de novo. So that means initially developed as a grade 4 tumor because of that multiple mutations, or it can be progressive like that. It is initially as a astrocytoma, then it is converted to anaplastic astrocytoma, then it is converted to that highest grade, the glioblastoma. So that means it is progressive or de novo. If it is de novo, different symptom. It is progressive, different symptom. And if it is progressive, early stage, some kind of things are appearing, and with stage, increasing stage, it is appearing the different type of symptoms. And many things are actually, it's when the brain tumors happens that other parts of the body organ, other parts of the body can be affected, no, based on that which nerve is affected. And because of that, one more thing is surgical problem, surgical resection is problem. Because that if the vital nerve, suppose facial nerve, if it is cut down, then what will happen? It will be there should be some problem.
I think there are some questions from students too. Trisha Chakraborty,
Yeah, Trisha Chakraborty.
Please switch on your mic and ask the question.
Trisha, you can unmute.
Hello, sir. Hello, sir.
Yes, please.
Sir, my question is so basic that if anybody is leading a normal life and looking like a normal person, but he or she has any kind of symptom in it that they can face a cancer in future. So how can we identify that? I just want to know that the physical any identification for knowing that the person is you will be facing the cancer in future or not.
Yeah, very hard questions. So I have to answer it. The hard questions, why? Because that what is not easily answerable, that is the hard questions, you know. Your question is how we can think about that if anybody can happens cancer, right? Today's lecture,
Yes,
I am not giving the depth of research. That means high depth of research, the high code molecular biological research. Just I gave the overview in that particular platform. because I want that at the basic things should know, we have to recapitulate in the awareness day. And if we can recapitulate those basic things, we can easily think about the future. And the particular thing that that clock is invisible, right? For everybody's mind, that I show the clock. And this is not the biological clock, that's the day-night clock. The biological functions based on that clock. No, not that clock. This is invisible clock. And this clock always ticks on in everybody's body. We don't know. That clock means it's a cell clock. That means all cells have the clock. And which cell the clock is stopped or clock is moving to another way, we don't know. So that means we cannot claim that this person will have cancer or not cancer. All of a sudden it can happen. And you know that in our body, every day cells are divided. Every day we are gathering many mutations, but all are not dangerous. So that means most of the mutations are not dangerous mutations. Those mutations can be cured or can be there, but that is non-functional, not changing any power of the cell. That is not they are efficient for transformation. Got it? So all the cells are actually getting this kind of triggering effect. So every day, every moment, something can trigger to our cells, but there is a fighting back mechanisms that can tolerate. So how long this fighting is winning by the normal cells, it cannot be. Suppose one cell is changed, but there is a possibility side by cell, like the immune cells, they are actually destroying that particular transformed cell. So it cannot form the tumor. So most of the events are preventing events. But when in some tissue somewhere, it is the transformed cell getting so much power, it can survive within the environment. By fighting, that tumor can appear. So you can see some people, lifetime they are smokers, huge strong smokers, but they don't have the cancers, lung cancer. But some people, they never goes to the bad environment, but still they have the cancer. So this is a very tough question to answer it, that who will have this particular disease? No.
Okay.
There is no technique available yet, no test available yet. So that means we can understand that lifetime.
No physically identification we can't get.
No, in a normal individual, no. Then everybody will go. Everybody will go and test it. That in lifetime I will have it or not. Because suppose today you are going to take the test, that test result telling that you will not have cancer in your lifetime. But tomorrow another mutation came and that mutation caused that cancer tumor. So that test is not valid tomorrow. Got it?
Okay, sir. Thank you.
But one thing is that genome sequencing is already done. So from the genome sequence, if anybody want to test for the genome sequence that in some stage, that genome sequence maybe through the artificial intelligence, one day can tell that there is a possibility. But it doesn't mean that it will be. No. But I hope that after another couple of decades, that artificial intelligence can tell the possibility of this particular disease can appear in individual's lifetime.
Okay, sir. Thank you, sir.
There's a question from Rupsha Laha. What is the role of mitochondria in cancer cell, and can mitochondrial targeted therapy be for cancer treatment?
वैसे तो कोई फर्क नहीं पड़ेगा, इतने से नहीं पड़ेगा, नहीं तो वापस कर दियो, ठीक है?
I think something is disturbing. Anyway, the questions I understand that mitochondria targeting therapy is possible or not. Yeah, it's right. So it's right, the mitochondrial therapy is possible. But the thing is that we have to be very careful that to identify the mitochondria in disease cells difference, the mitochondria disease cells versus the non-disease cells. And it is not an organ, it is not a tissue. It is within the tissue, within the cell, mitochondria is there. So mitochondria targeting is not so easy job, but it can be. Because if you disturb the mitochondria only to the target cell, then mitochondria will be damaged, so power will be gone, so cell will not survive in that particular environment. So if we can directly targeting the mitochondria, targeting drugs to the cancer cells only, then it's possible. Hello?
Yes, whoever asks a question like
Mitu Ghoda, I think, have some question. Ma'am?
Yeah,
please.
yeah. Thank you, sir. That was really enriching knowledge that you shared with all of us. My question may be, not related to you. When you have that liberty to say, no, or I'm a doctor. I'm an ophthalmologist by profession, and I am really happy about the research that is going on. My question is, we don't know which person is going to actually catch the cancer, as very correctly it was pointed out by Trisha also. And I am also an emotional fitness coach. I did my master's in emotional intelligence. And my curiosity towards emotional intelligence was that I always felt that it is the hidden emotions that cause lot of diseases, the separation of emotions. Because regarding in my practice also, I have seen that there are so many diseases for the reasons we don't know. And there are so much of recurrences of the diseases, which we don't know from where they are coming. So now that you're also a part of, research, a very big research in cancer, I have a strong feeling that the people who are of that type of personality, which are keeping their emotions inside, which are generating chemicals, which are ruining the chemical of our body inside and creating that environment of generation of the cancer. Is it related or not? That is my very big question. Because epigenetics the emotion part is because when emotions come, their addictions come, and then the whole lifestyle get messed up, the obesity, and it's all related to emotional imbalance, which show up as a physical, disturbance later on. That is what even I want to do, and I am actually I've done my master's in emotional intelligence for that reason, because I was very curious about it. Yeah.
Okay. Really, very nice. Very nice. Okay, Dr. Voda. So, very nice questions. And I thought that when you started these things, I thought that you will ask about the retinoblastoma and that the research perspective in the retinoblastoma and about the RB or the RB family proteins, how they are perspective. But you ask in the basic steps that emotional point of view, that emotions and the disease.
Yeah.
And we all know that stress and the disease.
Yes.
That means the emotion is very important for anything. And emotion can change the lifestyle.
True.
And the stress actually, that sometimes what happened that we all know that same medicine, but some people get out from the disease quickly. Some people get out from the disease very long time is taken. And some patients that same medicine is applied by the clinicians. Some patients are getting some adverse effect. Some patients, they are actually getting the sense of that very easily. So emotion is very much connected with everything, education, health, disease perspective, everywhere. But when the normal human being, they are under stress, there's a limited stress, there's no problem. So we all have kind of stress because of our emotions or different type of emotions, but this is temporarily. And temporary emotions cannot change us a lot. And because if you can correlate with some cancer in this particular platform, you can tell that breast cancer, prostate cancer, ovarian cancer, there's some cancers, those are the hormone dependent cancers. even the pituitary cancer. So hormone-dependent cancers and hormone production, hormone secretions, these all are connected with the stress and emotion. So that means that emotion does many things. And also I explained that in case of the prostate cancer, the first step that clinicians are actually taken, that is the androgen ablation therapy. So that remove the hormone from the systems. So that means hormone can change many things, and emotion is actually producing many such hormones, endocrine or exocrine, many such hormones, and hormones have various effects. So that means these hormones are temporarily produced, or temporarily overexpressed, or temporarily excreted, and give some effect. But when in the normal systems, when we are out of that emotion, that is again recovered, because we all have the balancing systems in our body. But in case of the emotion of a patient, that is different. The difference is that emotion regarding the life or death decision. Some disease, not all disease, and some disease like that, not life or death, but that recovery time. So that means that stress, that emotion is actually affecting the treatment system. That's all I can tell you.
Very true, very true. You're absolutely right. But honestly speaking, because I'm very curious about it and I observe people, the patterns of people. You have absolutely correctly pointed, emotion is a very transitory phenomena, right? And we, as a human, we have homeostasis, which balances out everything, right? But we have never been trained how to deal with emotions. Emotions suddenly change into feelings, and the kind of the resilience that we people have, and how we are trained to deal with resilience is not very appropriate, I would say, because the mindsets also decide how we behave, the choices that we make, and the lifestyle which we choose to live, and ultimately reflect into the diseases which we observe. This is, I'm saying, because of my, because my experience, that I'm saying, it's all by my experience, because the patterns I observe and how people, there's a more of fear rather than awareness. When we understand the disease from the point of fear, the chemicals that you generate in your body are different. And if we are understanding all the information only for the awareness and mindfulness, then the chemicals will be different. And that will decide the chemistry of our body and reflect our DNA and everything that's going to reflect. That was my main concern. And I always feel that it's important to create more and more awareness about how we can deal with the emotions mindfully, because the kind of diseases which are coming up, the emotional overload that we are experiencing, I think it's the maximum high that we're experiencing. So many of diseases which are coming up. And I still remember when I was doing my MBBS, there was a small column of cancer in our books. And now the amount of cancer that we are seeing, and I think the books have changed. There was just a small column at the end of any topic, pathology, that we used to study, a small paragraph about the cancer. So somewhere we have, I think, somewhere we need to work on that aspect also.
Actually, madam, like when the disease sets in, people get more panicky and more worried. I mean, even before that. So you are right. Like the emotions or like the stress increases, because now there are so many treatments that a lot of people are aware of. But if you think back, say a decade previous or even before that, it is once this word comes into a family, like the whole family dies, sort of the thing. So that is like, yeah, that is this awareness thing, that we need not be panicky and just take it, like you have said, that resilience, how it works. That is a very important factor, and we are very much happy from the organizers' point of view that we have one of the persons like you. So we have raised a different angle, have added a different hue to the webinar, I think.
Yeah, it is very important. And that regarding the cancer awareness, we know that this particular topic that she completed her MS and then MBBS. This particularly huge important topic, and in these days, this decades and onwards, it is very important, and we need it. And I can tell one thing, that when we were in school, that time there are some core subject like that physics, chemistry, mathematics, biology. Then the things are changing, that merging the subjects to understand better. So that means it's a merging like the biochemistry, biophysics, like that kind of different type of subjects appear. And recently, that happened, that the psychologists are there. The psychologists, they are actually treatment doing that point of view. And even nowadays, that the one subject is recently appearing, that is the, I think, clinical psychology, masters in clinical psychology. And that is very much important these days, because that people are busy. When you are more busy, then your brain is more working. So these more complications, that more functions. So for that reasons, we are getting more and more stress. And in that situations, if something unwanted events happen because of the boss or someone or the administrative issues, then this kind of stress or emotion appeared that is not favoring, but it is damaging the system. So that means this kind of awareness is more important, even more important than the cancer awareness. The cancer awareness is okay, but that clinical awareness, like the psychological awareness, is very important for not only cancer, any patients, for any patients. So that means like that in any hospital, there are some dietitians. So dietitians always telling that what kind of diet you have to take because you have that diabetes, right? So nowadays, the more important issue is the clinical psychologist in each and every hospital. So we need more and more clinical psychologists from master's degree and onward. They have to actually make the more awareness to the civil society. I think we need, and this is the need of the hour. Maybe we have to wait for another decade to get those kind of issues in each and every hospital. Like that one emotion, I can give an example, that insurance, health insurance. We all don't have the health insurance policy. So for the reasons, we are worried that how much money we can actually use for this treatment, and we all are not rich. Some are poor, some are mediocre, some are rich, just like that. So for the reasons that I think that kind of people need in the society, that not only goes to the hospital, but they have to, that awareness program, they have to start it on that particular psychological activities. And you know that year after year, you can see the societal psychological things are changing. It is changing every day. But if we very carefully look after that, every year, the people's mind are changing. So if you compare the 10 years back and 10 years after, everybody's mind change, psychology change. So psychology is very important issue. Another is the stress is very important issue. So that two issue clinically important. So we need those kind of people. Very much need. I don't know that all the countries have thought about that or not. So that means, ma'am, like you, every people who are thinking about the emotion, about the stress, or write something, bringing to the society that they have to read it, they have to claim it. So each and every claim, one day it can change. So that means it can be accelerated. Like that biotechnology in 90s, there is no biotechnology. In mid-90s, biotechnology came, and then what happens? Within a decade, each and every college started the MTech in biotechnology, MS in biotechnology, BS in biotechnology, then everywhere biotechnology. Then government is not thinking about their job. So that means that is a stress pathway. That means you are educated people in a particular area and you are not providing the job for them. So that is kind of stress automatically coming through the point of administrations from the government. This is another stress. And there are many people who are working in the IT sector. Recent days, we all understand how much stress they are carrying in each and every day. So you have to please write something, always write. So write to the newspaper, write to the system. So then you know others will be interested to write something. And even today's world is the Facebook and Instagram. Although I don't know those things, but Instagram, those things. So if you write, someone should write. So that means it will be propagated. And it is important.
It's very important.
Sir, students are very much curious. So there are two questions from students, I think. Yeah.
Do you have time? I have time. I have no problem.
Just a few, sir. One or two questions. Yeah. This is Shan Mondal. Yeah.
Sir, can I ask?
Yes.
Sir, I have the question that even after early diagnosis of cancer, still any further probability of cancer will be there? Sir, I am not asking on respect, I am asking irrespective of any cancer types. Still after diagnosis, early diagnosis, still any further probability of cancer will be there in the recent futures?
That is the early diagnosis means that you have some problem, you go to the clinicians checked all the possibilities and detected you do not have cancer. Okay? So
yes,
it doesn't mean you do not have cancers in future. Because I explained very nicely that all of a sudden it can appear. So because of some infection, you have a pain in your stomach or in your colon. Got it? And you go to the doctor, the doctor's actually doing the colonoscopy and telling that you don't have any problem. There's no tumor mass, there's no abnormality. Okay. So you are not worried about it, but after 5 years, you have again pain. And that pain is not because of the infection. That pain is because of that abnormality in the tissues. And that is the tumor is originated. Got it?
Yes, sir. Even after diagnosis, we can't guarantee it.
Nobody can guarantee it. That means you have a fever now and you are taking the medicine, fever gone. It doesn't mean within couple of days you will have fever again. Or you don't have fever in your future. Right?
Yes, sir. Thank you, sir, for the enlightenment.
I think the next question is from Shati Das.
I am audible, sir.
Yes.
Yeah.
Sir, my question is, sir, we know that cancer is genetic disease, but my question is, how can prevent it from spreading in the family?
I cannot hear properly. Tell me one more time.
Sir, we know that cancer is genetic disease, but my question is, how can prevent it from spreading in the family?
How can prevent it from spreading?
Yes, sir.
Okay. So that means there are some events that happens. That means cellular property. So that means that cells will go from one place to another place through a system, right? That system is the circulatory system. That means through blood, the cells can go from one organ to another organ, right?
Yes,
and one more thing is that inside the tumor, there is a huge cell mass. So for the reasons angiogenesis is there, that means new blood vessels goes inside the tumor, right?
Yes, sir.
Got it? And that is one factor is called the VEGF. That VEGF is important for this kind of invasion. That means their invasion goes to the blood vessel and then it goes to the other places throughout the body where it can go, it can sit and it can produce tumor. So that means you have to stop the angiogenesis process. So that means in the tumor, you have to produce the drug that can prevent this particular process. That means there are some particular events happening for the metastatic character when a cell can get. And that is ecadrin reduction, whereas encadrin, bimentin, those markers are increased. So you have to prevent your tackle with that. Encadrin and bimentin, those are responsible for kind of migration. If you can stop their production, you can stop the metastatic event. So the drugs have to be developed that can stop the production of these particular factors, those are important for invasion. So that when cell goes from tissue to the blood vein. You got the answer?
Yes, sir.
You
So
have
thank you.
discovered that it should not goes to the blood. It will be within the tissue environment.
Okay, sir. So
Sir,
thank you.
just a last question. I think there may be several questions. I think students or participants can ask later.
I think rest of the questions you all
Yeah.
can share. You all can answer them in future. Okay.
Sir,
Yeah.
there is another question from Dr. Reva Mondal. She's asking that as my question, what is the significance of AI for diagnosis and treatment of cancer? Just the last questions.
Significance of AI in diagnosis and treatment of cancers. Actually, if you think, on all of you, think in the direction that artificial intelligence not only for treatment and diagnosis of any disease. This is important for anything. So it means everyday life. This intelligence can be used by all students from school, college, university, everywhere. Here, artificial intelligence is a new subject, and this subject is acquired throughout the world, throughout the globe. And there are two things, not only artificial intelligence, the machine learning and the artificial intelligence. Two things together to give some answers, the plausible answers, not the right answer, at least plausible answers to ask whatever we are asking. Suppose you know that when you are using the game in your phone, then I think today's phone are that artificial intelligence making phone. So that means the phone is telling something. Suppose this is not artificial intelligence, that means you are speaking, and that is converted to in the message to goes to another place. Suppose in the TV, the today's TV, what happened? You were speaking that, okay, I want this channel, right? The AVP on the live. Then it goes to that systems and it is automatically on that AVP on the live. So all artificial intelligence. So that means that this kind of intelligence, when joined with the hands on the machine learning, of course, in case of the diagnosis and treatment, we have to get the data. That means each and every patient data should be recorded. So the patient's history, so each and every patient history will be kept as a recorded as a digital platform. Maybe the countries like America, Europe, those countries, they are recorded those things as a clinical record in their computer. So doctors should have a computer. But in India, in China, you cannot get that each and every doctor have to use the computer. So that means whatever the data we have in the big hospital or the good hospitals, the recent developed permanent hospital like the AIMS in India, the many private hospitals in US, Europe, there are many hospitals, Canada. So they are actually taking the report every day. So that means all the data is actually deposited. And then who is another organization, they are handling some of the data. And there is, the importance is that data mining. So that means from all these billions of data, that is the artificial intelligence that is actually helping us how we can mine the data according our wish. So that means I ask the TV remote that I need AVP on the live, so TV automatically goes to that channel. Like that in artificial intelligence that I need particular this disease for female at the age of 30 to 35, all the data will come. So that means this is a technology that whatever you want, it will come to you. Suppose the MRI data for the brain cancer, if you want that technology, that artificial intelligence, when it will be developed, that when you ask that, I want a particular type of brain tumor and the location and the age of and the sex, that all the MRI data will come. You got it? So AI is helping us, and in the future decades, I think we all understand that most powerful things in the world is the AI and ML, of course. But the thing is that we have to empower AI by our data. So we need to keep each and every data. We all know that things that we can, if you want to visualize your childhood, suppose one particular event you can memorize. So anytime when you were at the age of 5 or 7 or 10, if you want to recollect your memory, you can see that particular event. Visualize, right? That means that this is the power of the brain. And you know about the supercomputers, how much power it has. And that supercomputer was developed a couple of decades ago. But now the systems is highly upgraded. Now you can believe that robotic surgery, right? What happens in that? So that means artificial intelligence, should be nicely and properly maintained that in that particular subject area. So we need the expert. So this is a subject. We have to be trained enough manpower in that particular area. So that means many such good colleges should provide that artificial intelligence, masters in artificial intelligence. That there should be a domain of artificial intelligence, domain of the research domain of artificial intelligence. Then one day we can achieve that how much power it's in the diagnosis and treatment of any disease, not only cancer.
Thank you so much, sir, for patiently answering all the questions. And once again, thanks for that brilliant lecture and for your precious time. Thank you.
Yes, madam. When I joined this platform, I am for you. So that whatever you will guide me, I have to do that. So when the meeting is over, I am free. Otherwise, I am yours.
Thank
so
you.
Whatever you will guide me, I will do that. Okay. So now I am free, so right? I am free out now. So can
yes,
with permission, can I leave?
Yes, sir, of course. Thank you so much for your time.
Just a minute. We have to give the platform for Kirat sir for the vote of thanks, I think.
Yeah, right? No, I meant like
I have an announcement.
Like we're going to the valedictory session. That's like technically
just excellent session as expected, as always expected from sir. As we come to the conclusion of this enlightening seminar, I'd like to take a few moments to express my heartfelt gratitude to everyone who made this event possible. First and foremost, I'd like to thank our esteemed resource person, Dr. Mrinal Kanti Ghosh, for sharing his expertise, insights on cancer biology. Your presentation has not only informed us, but has also inspired us to think critically about the situation and possibilities of cancer treatment and the current scenario of cancer. Your lecture showed the current situation of different types of cancer, their dreadful prognosis, and possible therapeutic intervention, obviously, with the solution. Probable solutions. New technologies like nanotechnology, exosomes, nanotunnel, and robotics have been highlighted in your lecture, which may be the interventions of very recent future. I would like also to extend my sincere thanks to principals, faculties, and students of Department of Microbiology and IQAC of Raidiki College, Dhruv Haldar College, and Michael Madhusar Memorial College. Your hard work and dedication have been instrumental for bringing this seminar in fruition. A special thanks to Microbiology Society India for their collaboration and continuous support with us. Your commitment to advancing knowledge in microbiology and allied areas is truly commendable. Thanking all the students who will be participating in poster session competition for your creativity and effort in advance. You are encouraged to participate in the poster session. The conditions and the deadlines will be announced just a few seconds after this session. Your contributions are vital for advancement of understanding in this field, and this type of sessions are only for you and other parts of the society. Finally, I'd like to thank all the attendees for your participation and engagement. Your presence have today's reflects your commitment, learning, advancing in the field of microbiology. I would like to also thank, as a state president of NDSI, Dr. Malini Ghosh and Dr. Rudima Vishwas for their continuous support and for making an idea of this type of seminar. Let us carry forward the knowledge and we have gained today, and continue work towards a future where we can make significant strides in cancer research and treatment. Thank you, and have a wonderful evening. Thank you.
Okay, thank you all.
Thank you,
Let me leave.
Yeah.
I have to go to home.
Thank you so much, sir.
Thank you, sir. And thank you, Kirat, sir. So now I'll present my screen and share information on the competition. So is the screen visible?
Yeah, it's visible. It's visible.
Okay, so these are basically the guidelines for students' poster competition, as we have already circulated. So the theme is microbes and microbial products in cancer treatment, the recent insights. So students, let me point out that this competition has been arranged so that you will go back to textbooks. You will also consult e-resources, please, not just Wikipedia. Okay, go for recent research papers and go through them. And based on the knowledge acquired, you prepare your poster. And most importantly, through your learning, whatever awareness you are getting, please share it with your family and friends and the neighborhood people. That is most important. Okay, so coming to the technical parts, posters have to be prepared digitally. Both UG and PG students of microbiology, biotechnology, and botany can participate. There will be separate submission links given for UG and PG students. Please remember that one student can submit only one poster. Now, posters can be submitted individually, like by a single student as the author, or it can be a teamwork, but a group of maximum 3 student authors, okay? And the corresponding author should be named separately. The poster should be based on original articles within the broad area of the above mentioned theme, but do not just copy paste, okay? The links will be given this Sunday. Do not leave the Telegram group. It will be shared in the Telegram group. You can submit the posters in PPT or PDF form. Submit by 17th November 2024 midnight. Positively by that time, on that date, otherwise the link will be inactivated. The posters will be screened by experts, and the first 3 best posters will be declared as winners, and the authors of those posters will be presenting their posters in on a date which we will announce in that same Telegram group later on. And on the same day, all the other posters will be also shown on that platform to encourage more participation from you. It's not always about winning, you know. Please remember that your poster must contain the following points. It should have a title, name of the authors, name, email ID, and phone number of the corresponding author. It should have an introduction, that is the background, objective, results or your observation, and conclusion or the key outcomes of your poster. And add a list of references, at least 3 references. So I hope that was clear. This has also been shared with you in the Telegram group. So I hope you all enjoyed today's lecture, and with that I'll hand over to Kirat sir for declaring the seminar end.
Yeah, this is the conclusion of this today's session. All the students and participants from all over India are encouraged to participate in the poster session. We hope a good number of posters will be submitted. There will be enough time, and by Sunday we will provide a link in the Telegram group where you can fill up everything and upload your respective poster. It will be better if you prepare the poster in the PPT and then convert it to JPEG format and upload in the link. Okay. And for further clarification, please communicate over the Telegram group. So that's all for today. Thank you all. Thank you, madam, Urlima madam, and Malini madam for developing such a beautiful session. Thank you.
Thank
Yes,
you, and
thank you,
good night
and
to all of
good
you.
night. Yes.